So in today's experiment, we are going to perform the microscopy and transverse section of another important traditional medicinal plant that is known as Rauwolfia. So as the biological source indicate, Rauwolfia obtained from the dried roots of Rauwolfia serpentina belongs to the family Apocynaceae. So it is mainly used as anti-hypertensive drug to reduce the hypertension. The root of this drug is used traditionally. It mainly contains alkaloids, indole alkaloids like reserpine, ajmalin, yombin, etc. This root is also known as sarpagandha. So you can see here the root shape is like snake movement. That's why it is known as sarpa. Sarpa means snake. So due to this movement, it looks like snake movement. It is known as the sarpagandha root. Let us see how to take the transverse section. This root is very hard. You can see it's not breakable easily. So we need to boil it at least for 1 hour to 2 hours so that all the cellular contents get softened. Then only we can able to take the transverse section. So for this purpose, we have already our previous boiled sample. Previously boiled sample what we have kept here. This is the softened one, and we are going to perform the transverse section from this section. Let us see how we are taking the transfer section. So first thing, with the help of a blade, you just cut it. So you can see the surface is level now. Now with the edge of the blade, you take always take the section so the edge of the blade, so that your section will be thinnest one. So if your section is thick, then you may not observe the cellular characteristics properly. Remember one thing, how to select or how to know which one is the thinnest one or thickest one based on the color. Lighter the color, thinner the section. Darker the color, thicker the section. So from the various section, we have to choose the lightest color section, which is the thinnest one. So like that fashion, you hold it tightly with your finger and nails. Then through the edge of the blade, you try to take few transverse section in this fashion. Now transfer this into distilled water and soak it for some time. It's already soaked. Now from this, you choose which one is the lightest one. You transfer 1 or 2 such sections over it. Now next step is the addition of reagents. First you add 1 or 2 drops of chloral hydrate solution. You can see this is the chloral hydrate. So I have already prepared the reagents with the chloral hydrate. So we are adding the chloral hydrate here. What is the use of chloral hydrate? Chloral hydrate is used as cleaning agent. What it cleans? It cleans the pigments, earthy substance like sand, soil or any other adhering substance to it. You place it over it. Now add 1 drop of fluoroglucinol and 1 drop of concentrated HCl. This is the fluoroglucinol solution. So 1 or 2 drops you just add, and just add 1 or 2 drops of concentrated HCl. Mix that properly. Wait for some time. What is the purpose of adding fluoroglucinol and HCl 1:1 ratio? It stains all the lignified characters into pink color. So just wait for some time so that your cellular character stains properly. Now you put the cover slide in that fashion. While adding the cover slide, very careful, it should not contain any air bubble. If air bubbles are present, it may hinder to observe the cellular characters significantly. Moreover, if the air bubbles are present inside the slide, then you remove the cover slip once again and place it. Now our slide is almost ready. Now you wipe out excess of reagent with the help of a filter paper. You blow it out and then you place it under the microscope. Focus at 10x and observe the cellular characters. So actually what you are going to observe here, so as it is a root, first you will observe the cork. Generally cork appears to be brownish color, but in this case the Rauwolfia cork is a very specific character. It is known as stratified cork. Due to the it appears slightly pinkish in color due to the presence of suberin in that. It appears pinkish in color. Suberin and lignin present in that, it appears pinkish in color. Followed by you will find the phellogen and phelloderm. Phellogen and phelloderm are merged with each other. And to the cortex part, you will find n number of wood parenchyma. Then you will find secondary phloem, wood fibers. All these things are lignified characters, and you will observe very distinctly in the transverse section of Rauwolfia. So in brief, what you are going to observe, first thing you will find stratified cork, followed by phellogen and phelloderm, which are merged together. Then you will observe in the cortex region, you will find the vessel, wood parenchyma, wood fibers, secondary phloem. All these are lignified characters you will find. So in brief, these are the microscopical characters of Rauwolfia root.